Good morning, bio students. I wanted to make a video sort of highlighting what you got going on Monday and Tuesday of this week. I will be gone for a few days. So you can go find in Canvas, go to our daily lesson 131 and 21, sorry. Make sure you write down your root words. Make sure that you go through and take your quiz over monohybrids. Just wanted to give you guys a short quiz looking at monohybrid crossing, making sure you understand the terminology. Based on that CFA I had you guys take last week, pretty good mix. A lot of you are doing well. Some of you, I think, just need a little more practice, but I think we're all moving forward, which is good. Make sure you watch this video. Hopefully you're doing that. This is going to explain how to do the next sort of inheritance that we're talking about, which is incomplete dominance and codominance. And lastly, complete pages 5 through 7, your genetics packet, and then anything that you have left over from 1 through 4. So let's not take any more time. I want to get into this. Let's look at, make sure I can mirror my screen here. Let's look at how to do some of these incomplete and codominant problems, okay? So as we look at what incomplete dominant is, it's essentially when an organism inherits two different traits and neither is completely expressed, okay? So when you inherit two separate traits and neither one gets completely shown, okay? And I always think of it as sort of a meeting in the middle, all right? And you get some sort of phenotype, or the individual is going to look somewhere in between what the originals look like, okay? So it looks like meeting colors, essentially. So let's say red is a trait, white is a trait. Instead of the offspring being red or white, you get something new, like pink. And so this is one of the only times where we get a new phenotype based on our genotypes, or brown and white make tan. And so we're going to go through a few examples here together, and I'll show you how I want these set up. Again, you've seen I'm kind of picky on how I want these done, and I think as long as you can do it this way, you'll be fine. So a lot of traits are this way. I wish everything was just regular dominant recessive and followed those rules, but we talked about some of the wrinkles in those rules, and that we're continuing to build on our understanding of genetics. So notice that I'm using the same letter. So in here we're looking at flower color, and I like the visual, but I just don't like I don't necessarily I don't like the color or the way they wrote these. So we would typically not see it written as big R, and big W, big W. That indicates two separate traits, and so we would not write it that way. Instead, what we would do is we would use a letter because it's the same trait, flower color, okay? So we might use a D to represent flower color, and then we might use a capital D with an R to represent dominant red flower color, and a capital DW to represent dominant white, okay? So we have to write these superscripted letters. And so it says, cross a homozygous red flower, DR, with a homozygous white flower, DW. So I've already given you the genotypes. This one goes here, right? And then this one goes here. And so we set up our Punnett square, right? We get the red flower over here, white flower over here, and in every single box, we get a W and an R, right? Every single box in our Punnett square, we get one R and one W. So you get a red allele and a white allele. And because this is incomplete dominance, they both sort of meet in the middle when you get the phenotype. And so we would write this as this. We would say 4 DR DW, and because this is incomplete dominance, the red and the white mix to make 4 pink. Okay, let's try another one here. So what would we get if we cross one of the F1 offspring? Basically, if we just took one of those pink flowers we just got and crossed it with a white flower, well, we would get a Punnett square that looks something like this. So we're going to cross one of our F1s. Okay, so that was a pink flower, which was DR, DW, one red allele, one white allele, and a white flower, which is DW, DW. We get DR, DW, 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 another DR, DW, and lastly a DW, DW. So now, before it was, you always write the most dominant genotype first. Well, there isn't really one that's more dominant, so we tend to write it alphabetically. So we would write our two DRs first. I've got one there and one there. So I'm going to say for my genotypes, I'm going to say 2 DR, DW, and 2 DW, DW. Well, how are those flowers going to look? Well, the first two, one red allele, one white allele, makes 2 pink. And DW, DW, white and white makes 2 white. And that kind of makes sense. You cross a pink flower with a white flower, what do you get? You get more pink flowers, you get more white flowers, and that makes sense, right? But what if we cross 2 pink flowers? Let's take a look at that. Let's cross 2 pink flowers down here. Pink flowers had one red allele, one white allele. We get DR, DR in the first, DR, DW in the second, DR, DW in the third, DR, DW, DW in the fourth. Okay, so now what do we get for genotypes? Well, I'm going to write them alphabetically. So I'm going to write R comes before W in the alphabet. So I'm going to write my DR, DR. Looks like I've only got one of those right there. Then I'm going to write my 2 DR, DW. Fix this quick. And then lastly, my 1 DW, DW. So here I cross 2 pink flowers, and what do I get? Well, we're crossing 2 pink flowers, and we get 1 DR, DR. So 1 red flower, 2 DR, DWs, so 2 pink flowers. And lastly, 1 DW, DW, or 1 white flower. So this one's kind of interesting. You would think in crossing 2 pink flowers, you would get more pink flowers, but you don't. By crossing 2 heterozygous individuals in incomplete dominance, you actually get all possibilities. Crossing 2 pink flowers can make red flowers, pink flowers, or white flowers. Now, we know that we have a 2 out of 4 chance, or a 50% chance of those new flowers coming out pink. But you also have a 25% chance of them coming out red or white, okay? And so let's try an example looking at horse coat color. Okay, this is a Palomino horse. Palomino horses, they have sort of this tan coat with sort of this stark white or stark blonde hair or tail, mane, and so people people tend to like these, and so sometimes people will specifically breed for Palomino horses, and this is essentially how it works. This is if you wanted to make Palomino horses, this is how it's done. And so in horses, the color of their coat can be caused by incomplete dominance because there is more than one dominant gene for coat color. Chestnut or brown coat color and cream white coat color are both dominant genes, and Palomino, which is sort of a tan, is usually the result of incomplete dominance in the crossing of a chestnut and a cream horse. Use the key provided and cross 2 Palomino horses, showing all your work, including percentages and ratios. Well, let's do that. So this CH is chestnut. So if you get 2 CHs, remember I'm doing a coat color and a coat color, and I'm using a capital H to represent the dominant chestnut allele. And then I've got cream, still using C, but the R is going to represent that dominant cream allele. Okay, so if you get 2 chestnut alleles, you're going to be a chestnut horse. If you get 2 cream alleles, you're going to be a cream horse. But what if you get a chestnut allele and a cream allele? What do you get? Well, you get this new thing. You're not brown, you're not white, you're sort of in between this tan coloring. Okay, and so we're going to cross 2 of these guys, 2 Palomino horses. CH, CR, CH, CR, we get CH, CH in the first, CH, CR in the second, CR, oops, CH, CR in the third, and CR, CR in the fourth. Okay, so our genotypes, H comes before R in the alphabet, so we're going to go with our 1, CH, CH, our 2, CH, CR, 1, CR, CR. Oops. Oh, it's wanting to, here. Phenotypes. We get 1, CH says chestnut, CH says chestnut, so 1, chestnut, CH says chestnut, CR says cream. You mix those together in incomplete dominance, you get 2, palomino. And lastly, you get 1, oops, that's another R, 1 cream. So if you mix 2 palomino horses, you actually have a 50% chance of the foal or the baby being palomino, right? And so as you know, you talk to ranchers or people that breed horses for a living, they're very in tune with the genetics of the animals that they're raising. People pay very close attention to this because sometimes, the results aren't obvious, right? You could mix 2 palomino horses and that the foal you get is actually going to be more of a darker brown color. And so that may or may not be something you wanted. And so paying attention to those genetics is important. And so I ask you a couple questions about that on your assignment. Make sure you peek at that. Okay. That's incomplete dominance, sort of a meeting in the middle. Neither one completely wins. You can see that here. Chestnut didn't win, cream didn't win. When you put them together, it makes something new. We also have something else called codominance. This is where both dominant genes, both now, are going to get expressed. So imagine if you have, white and red, those don't come together to make pink, right? What you get instead is sort of this red and white sort of patchy coloring. And you've seen this in animals. Most commonly is where we see it. So, whether it's, horses that are paint horses or cattle that have sort of that coloration, that sort of, two-tone to them. We see this pretty frequently in animals. And so we wanted to talk about this a little bit. This is codominant. So in this case, well, what happens if white is dominant and red is dominant? You don't get pink. You get red and white both being expressed. You get parts that are red and then parts that are white. Okay. So let's look at an example here. So we're going to look at codominance. Notice I'm going to use the same letter. I can't use 2 different letters because that indicates 2 different traits. I'm going to use F for feathers. Okay. We'll use F for feathers. And so black feather colors is FB and white feathers is FW. And both are dominant and display codominance. And this will be the trick when you take your quizzes or you do an assignment. You need to pay close attention. Is the trait codominant or incomplete dominant? That will change how you interpret your phenotypes. So be careful there. We're going to cross 2 spotted chickens. So in our example here, we crossed a white feathered chicken and a black feathered chicken. And what do you get? Well, instead of getting gray like you would have in incomplete dominance, you get chickens that have both black feathers and white feathers because they have both of those allele. They have a black allele and a white allele there. Okay, so you get black and white both being expressed. They're both dominant. They both want to be expressed. Okay, so, but what if we crossed, it says 2 of these spotted chickens? So we want to know, what if we cross 2 of these guys? What would we get? Well, we would get feathers black and feathers white, and another feathers black, feathers white. We get FB, FB in the first, FB, FW in the second, FB, FW in the third, and FW, FW in the fourth. Our genotypes, B comes before W in the alphabet, so we're going to go 1, FB, FB, 2, FB, FW, You can see them right here. There's one, there's another one, and one FW, FW. My phenotypes then, black feathers and black feathers, you're going to get one with black feathers, black, oops, black. Boy, that's not going well. Let's just fix that. One with black feathers, FBFW, codominant now. Before, we mix the colors together. Black and white would have made gray, but now that's not what happens. Both want to be expressed. Black doesn't want to back down. White doesn't want to back down. We get two that are black and white. We'll just say spotted, even though it's not really spots. Okay, and then we get one white. So again, little difference, same concepts though. The biggest thing is knowing how to write your alleles. Instead of just writing black and white, that indicates two separate traits. Don't write it that way. We're going to say that our feather allele can be black or white, indicating those are both dominant. We can't use lowercase letters here. It's codominant, okay? And you may not be too interested about, chickens or what color feathers they're going to have, but something that's probably, relevant to you or will be eventually is blood typing. Blood typing is actually a codominant trait, meaning there are different alleles that are dominant and neither of them backs down. And so there's really two proteins in your blood cells, okay? I better use a different color here. Use purple, okay? So this is a blood cell. You can see up in the right there. Blood cells either come in, have an A protein. So let's just kind of go like this. We'll say this is an A protein. We'll draw it as a little there. And if you remember, on cell membranes, there's receptor proteins. That's how cells talk to each other. Your blood cells also have those proteins on the outside of them, okay? And so if you have an A protein on your blood, well, maybe we'll represent it with a little triangle, purple triangle, right? You would be type A. If you had, let's say, let's use a different color here. We'll use green. We'll make a little square here. If you have this protein on your blood, the little green square, oops, you are type B. So if you had that, you would be type B blood. But what if you have both? Well, because they're codominant and the B protein is present, the A protein is present, you would actually be called type AB. So that's a blood type. You can be type A if you have the A protein, type B if you have the B protein, or you can be type AB if you have both. But what if you don't have any? What if there are no proteins on your blood cells? Well, then you are considered type 0, okay? Or type O, we say, all right? So you really have four possibilities. A protein, type A, B protein, type B, none of them, type O, or you have both of them, which is type AB, okay? And there's two ways to write this, and you'll see this when you get into your assignments here. You can write it as IA to represent A. It stands for immunoglobulin A. That's what that little protein is called. Or you can write it as just an A. And so we say that A and B are both dominant and O is recessive. Well, O is nothing. And so if you have one of the A or B proteins and you compare it to nothing, well, then you're going to show whatever that protein says. And so we're working with both sets of rules now. We're working with our codominance between A and B, and then our dominant recessive rules with the A, B, and then comparing it to O, okay? So let's look at an example here. I think it's a little tricky to see these things, and so let's look through one. We'll grab some pink here. So for you to be type A, right? Let's say you're type A. You could have gotten an A from each parent to be type A, Or you could have got an A from one and an O from the other, right? If O is nothing, then your body's going to code for whatever A says. It's just that like dominant recessive trait. And the same thing for B. You can be type B if you have two Bs, or if you have one and then nothing. The only way to be type O is you have to have two O's. As soon as you get an A or a B, those are going to become dominant and you're going to have those A or B proteins on your blood cells. Okay. Now, if you get an A from a parent and a B from a parent, you're going to be type AB because they're both dominant. All right. You've also heard of this thing called the RH protein. There's another protein on your blood cells, and if you have it, you are positive for it. And if you don't have that protein, you are negative for it. And it follows the same dominant recessive rules. Okay. So if I wanted to show somebody who was A positive, I would give them the A protein. I think that was a triangle. And then I would give them the RH protein. Let's do that as a little Y. So this would be somebody that's A positive. What if I wanted somebody that was B negative? I don't like that color. I wanted somebody that was B negative. Oops. B negative looks like this. I think we used a little square for B. And notice I'm not going to draw an RH protein over here. They have the B protein, but do not have the RH protein, making them negative. Okay? And so this matters quite a bit, actually, when you're trying to figure out who can donate to who, right? Because the thing is this. If, let's say, and I'm actually A negative, that is my blood type. If I'm A negative, my blood cells look like that. But I have these little antibodies. They're called floating around in my blood. Okay. And I have the B antibody. And what these antibodies do is they will lock on to any cell that is carrying those B proteins. So let's say you gave me blood with that B protein on there. When my body's look and recognize these cells, if you were to give me, let's say I was in an accident or something like that, I had to get a blood transfusion. If you were to give me blood cells with these B proteins on there, my antibodies would flag it. They would lock onto this thing, flag it, almost like a little red flag. And it would tell my immune system to go destroy these cells. Now, this is super helpful. Your immune system does this. That's how it figures out if you're being invaded by a bacteria or virus. It basically says, hey, I don't recognize this new cell with this new protein. Let's get it out of here. Okay. And so you can get really, really sick if somebody gives you the wrong blood. Okay. Let's take a look at, and we'll come back to these. Let's take a look at this chart briefly, and then we'll be done for our notes here. Notice, as you look at who can receive what blood, who can donate what blood, let's talk about why. Okay. So as we look at our blood cells, right? If your blood cell is O negative, let's say your cells are O negative, they don't have any proteins, right? Your blood cells don't have the A protein, the B protein, the RH protein. And so your body has never seen an A, a B, or an RH. So if you were to give somebody, this is recipient, you give somebody that's O negative, you give them blood. If it has an A protein, they will reject it. Let me change colors here. They will reject it, right? I won't use green. Even this RH, somebody who's O positive, they'll reject that blood too, okay? B protein, they'll reject it. They have never seen an A protein, a B protein, or an RH protein. So they have those antibodies, those little flaggers, to lock on to any of those cells. Okay? So if you are O negative, the only blood that you can get is from somebody who's O negative. Okay? But if we're talking donors here, if you're O negative, you can give to anybody. Why? Well, there's no proteins on there. There's no A, there's no B, there's no Rh proteins. There's nothing to flag. And so if you are O negative, your blood is very valuable because it can be given to anybody. It's called the universal donor. Now, we call AB positive people the universal recipient. Notice they can get blood from anybody here. Why? Well, they already have the A protein. They already have the B protein in their blood. They already have the Rh protein. There's nothing that you can give them that they haven't seen. They'll recognize everything, whether it's an A, a B, or an O, from anybody up here. They'll recognize that as somebody who's supposed to be there. Okay? And so make sure that you have some understanding here of who can donate to who can receive from who, and also why, right? It all comes down to those little proteins on your blood cells, okay? And the body's either understanding and recognition of those or lack of understanding and recognition of those, okay? So that's where I want to end the video. Oh, I think I actually do want to go through a couple examples. So let's cross a homozygous type A with a type O here, okay? Practice a few examples so you kind of know how to look at these. So homozygous type A, make my little Punnett square. Homozygous type A would be A with a type O. Well, the only way to be type O is if you have two of them. As soon as you get an A or B, that's what you are. So what do you get in this first box? You get an AO, another AO, an AO, and an AO. So if somebody was homozygous A and somebody was type O, this is what you would get for genotypes. You would get 4AO. And phenotypes, what would your blood type be? Well, you can have A and nothing. Well, A is going to dominate the O because O is nothing, and you're going to end up with 4 people that are type A. Let's cross somebody who's heterozygous type A and somebody who's type O. Well, the person who's type O is still OO. The person that's heterozygous A, this means you're going to be A, right? Heterozygous A would be AO. You're still type A blood because of the A is dominant, but you're heterozygous, meaning you have a different letter, okay? And so in this first box, we get AO, and then OO, AO, and then OO. So for genotypes, you can get 2 AO and 2 OO. So 2 type A, remember A is dominant, and 2 type O. You'll notice we are ignoring the Rh factor. We're not doing A positive, A negative, O positive. That would create a dihybrid cross. And so we're going to stay away from that, at least for now. Okay. So we're going to cross heterozygous A and heterozygous B. Heterozygous A would be AO. Heterozygous B would be BO. We get AB in the first, BO in the second, AO in the third, and OO in the fourth. Okay. Well, we get over here for genotypes, we get 1 AB, 1 AO, 1 BO, and 1 OO. So if you cross 2 heterozygous people with type A and type B, you could get really any blood type, right? You can get somebody who's type AB. You can get somebody who's type A, right? That's AO. Somebody who's type B, and then you can get somebody who's type O. So they used to try and use blood typing to help with paternity cases to try and figure out who the father is. And it's not always accurate. And you'll see why. Okay. So let's say the blood type of a mom is AB and the blood type of the baby is BO. What must the father's blood type be? Well, if the mom if the mom is AB and the baby is BO, we know that mom passed down the B, otherwise that couldn't be there. Okay? So, if the mom is AB and the baby's BO, I think, right? The dad is going to have an A, right? We know that. Otherwise, where did that A come from? Because the baby's sorry, I messed that up. Let me change this. I got too much writing going on here. If the baby's BO, we know the O had to come from dad. Okay? So mom's AB, baby's BO. We know the B came from mom. Otherwise, she would have passed down the A. But what could dad be? Well, dad could be AO and pass down the O. He could be BO and pass down the O. Or he can be OO and pass down the O. So dad could have been type O, type A, or type B and still created a baby that is BO, right? Again, a tricky there, okay? And then we see the same thing down here. If the mom is AO and the baby is AO, what could dad be? Well, if mom is AO and baby is AO, mom could have passed down the A or the O. We don't really know. And so essentially what you could get, I don't have any more room over there, right? Let me just change. I'm going to change colors here. So mom is AO and baby is AO, right? What could dad be? Well, he could also be AO. Mom passes down the A, dad passes down the O. That works. I think dad could be AA. Mom passes down the O, dad passes down an A. That still works. I think dad could be a BO, right? Dad passes down the O, mom passes down the A, you get AO. I think dad could also be AB. Dad passes down the A, mom passes down the O. That still works. And dad could also be OO. Literally, the only thing dad couldn't be would be BB. This is the only thing he couldn't be. And so again, we don't always use, well, actually, we don't use blood typing for paternity. We use, DNA analysis, things like that. And this is why. Sometimes it can eliminate a potential father, but it has a really hard time figuring out who the actual father is. I mean, you still have 5 possibilities there left over. And so that didn't help. It maybe eliminated one, but didn't give much further insight in there. Okay. You'll have a little activity on this as well. And I think that'll be one that is a good one for you to do on blood typing. It's kind of fun, and hopefully when I get back, we can talk more about this. Just want to make this video explaining how to do some of the work in your packet. Good luck, and we'll see you guys soon.